Um, yeah, so essentially, if you look at these types of images, the images on the left have contrast, and so it's to visualize your vessels. You can see the arrows pointing to the white thing that's a vessel here. The other on the right side is without contrast. It's more of an anatomical scan, and you can see the stroke is happening right where that vessel stops providing blood flow. And on the other, in this mirror image over there, the non-contrast one, you can see there's a hyperdense region. That is the blood clot itself. And so the blood clot that is causing the stroke is typically made up of a bunch of different blood components. This is red blood cells, it's fibrin, it's platelets, it's some white cells. And this material, how we treat stroke, is either broken up by a drug that we call TPA, or what's more and more being used and was actually pioneered here in the neurosurgery department is the use of mechanical thrombectomy. So that's either aspiration catheters that they navigate from your leg all the way up to your brain to suck out this clot. Stent retrievers where they kind of deploy the stent within that clot mass and pull it out, or kind of combination approaches, right? And so we've seen over the years that mechanical thrombectomy, or MT here, has much better outcomes. But the success of thrombectomy is dependent on how fast you can achieve reperfusion, meaning that if you can go up there with these devices and pull out the clot on the first time, you have a much higher rate of success and the patients do much better. And this is something that we call FPE, or first pass effect. And so we really want to achieve first pass effect. But that doesn't always happen. That only happens in about 30% of the cases. So only 30% of the strokes that are coming into the hospital are achieving the optimal outcome, right? And why is this? Well, this largely depends on the interaction between those devices and the blood clot that is in the brain. And so this is just a smattering of different histological staining of some of the clots that we've retrieved from patients. Back in 2018, I was part of an effort to set up a biobank of this material. And this is kind of the cellular composition between the fibrin, red cells, and white cells in the first 50 cases that we recruited. And you can see it kind of spans the board in composition. So they're extremely heterogeneous. Sorry. When we look at how this biological makeup could be affecting this outcomes, we do see that the first pass effect is highly correlated with the concentration of fibrin in these clots, right? So it kind of makes sense. The stiffer the clot, the harder it is to retrieve by any mechanism, right? Unfortunately, other imaging parameters like hyperdensity on the CT imaging, or something that we call perviousness, which is derived from both of those types of CTA, were not actually very indicative. They had very low ROC levels in terms of predicting whether a patient would have first pass effect. And that kind of leads to a conundrum. How can we assess first pass effect, or the propensity to achieve it, if we have to take out the clot to begin with, right? Not to mention there's other properties in these clots like nets, or these are essentially activated neutrophils that also provide further structural rigidity because of the DNA that they release into this clot. And you can see here's evidence that these other biological phenomena like nets are also associated with worse outcomes. And so we have a pretty good picture from the biological standpoint of, oh, what is associated with poor outcome and lower rates of this, fast reperfusion. And so with that understanding, we also have industry that has now started to tailor the thrombectomy devices that they're making towards specific types of clots. Some are made to aspirate larger weaker, structurally weaker clots. Some are made to go in and get really tough calcified clots. And so, we're at an interesting point where we are really kind of tailoring the treatments for personalized medicine in stroke, but there's a big gap. We don't actually have any biomarkers to detect what type of clot this is, right? What treatments will be more difficult? Is this a very tough clot? And so that has been the focus of my research for the past few years. And how we've gone about that is by the use of imaging radiomics, right? So I said, some of the very simplest imaging parameters haven't really tracked well with procedural success, but the biology of the clot really has, as evidenced by the body of literature so far. And so what we have been trying to do is find specific textural patterns in the CT images using radiomics that are associated with certain types of biology and then are predictive of outcomes. And not just outcomes, but all sorts of other parameters that a stroke surgeon or neurologist might be really keen to know, like whether this patient will improve quickly, what type of stroke was it, all that. And so this is kind of the main gist behind the research, is identifying specific radiomic patterns in the imaging, and then kind of understanding those by comparing it with the biology and then using those in predictive models to predict outcomes. And this is, for our pathology department, this really falls in line with kind of the guiding theory of the computational pathology as it stands right now, that what we can try to understand from the phenomenological level or the radiomic level is data and from underlying layers of information. So we're trying to understand data in the histomics and histological level, and then gene expression level, and then even down to the genomics level, just from the imaging. And that is a very challenging task. But we've shown that when we have really high resolution micro CT, so this is basically the same type of imaging technology, but just extremely longer scans in a resolution of 5 microns, almost subcellular resolution, we do see in the same plane for the histology and the radiology a very almost one-to-one correlation between the biology and the intensities that you see, which kind of proves the point and gives us belief on what we're doing here. Sorry, I hit the wrong button. And so, given this premise, we've published several studies that are linking these radiomics to certain biological phenomena to outcomes. We've been pretty successful at doing that. And we've kind of come up with this whole idea of how now we can translate this into the clinic, right? And so we've kind of come up with, or I have imagined this, this software that interacts with the imaging suite, right? So a patient would come in, they get their CT scan for their CT stroke study, and then before they even make it to the interventional suite, which is done under a different type of fluoroscopic guidance, there is a pipeline that takes this image, it identifies the clot in the CT scan, it segments it, it calculates the radiomics, it uses that and the patient's data to then give predictions about the stroke that it can then feed to the clinicians, and maybe they can tailor treatment or they can do appropriate patient triage. And so that's what we are envisioning with this whole research project. But the one big challenge has always been clot segmentation. And this is an incredibly difficult challenge because clots are extremely tiny. And in the whole image that is taken of your brain, it's a small fraction of a percent of the volume, right? Not to mention they're low contrast, so there's There's low signal. They're, like I said, extremely heterogeneous. So you can't just segment that region based on a single intensity threshold. They tend to be extremely small, and the anatomical complexity of the brain, where there's vascular tissue, brain tissue, and skull tissue, makes it very difficult. And so even expert surgeons and neurologists and radiologists disagree almost half of the time on where this clot is. And so we thought of basically using deep learning to help segment this, right? And kind of overcome this bottleneck of having manual intervention have to look at this and segment it, which would take, hours and would be well beyond that time treatment window, right? So in a study that we're kind of in the midst of publishing and working through the IP issues, we took about 223 treated stroke cases that came to our hospital. We have both CTA and non-contrast CT from them. We co-registered those on top of each other using the skull as kind of a rigid region to co-register these two images. We then manually segmented the clots and kind of took a consensus of a couple different segmenters. And these clots were all of different sizes and different hyperdensities. We then trained a deep learning model, which was a 3D full resolution NNUNet that had a 5-fold cross-validation. And the other interesting thing that we wanted to do is because we knew that the challenge of segmenting these clots has a lot of uncertainty and even, experts disagree, we tried to introduce a metric to quantify the uncertainty in segmentation. And so we use this kind of trajectory-based posterior sampling that had been published in the literature. And essentially what it is doing is it creates, using different initialization points, slightly different models to segment. And then based on, how the consensus of the model in terms of where the clot is, it then assigns an uncertainty score. And I'll show you a little bit of that in a little bit. Once we had this model with the uncertainty quantification, we then validated it in almost another 100 cases, comparing the segmentations to ground truth and looking at the uncertainty calculations, and then evaluated it kind of in the standard methods to evaluate kind of overlap of 3D objects. And so this is a couple examples of how well this segmentation and then UNet is working. You can see the green area, I don't have the laser pointer, but the green area is the ground truth, and the orange boundary is the ML prediction here. And so you can see we had excellent performance. The overall dice or the dice similarity coefficient was almost 70%. If we look at just the surface dice, it was much higher than that, which is really good. This algorithm performed much better in the hyperdense clots, which kind of makes sense because those are the ones that you can see easily. They also performed, much better on the medium and larger clots because, those are also easier to visualize on the CT imaging. And you can see most of the predictions were within two standard deviations based on these Bland-Fulton plots in terms of metrics like, mean volume. This is a plot of the mean volume. When it comes to the uncertainty, you can see here in this example in case A, the predicted area actually contains regions that are beyond the ground truth. And interestingly, that is the region that is highlighted the most in terms of the uncertainty metric. And so the uncertainty or metric that we call U clot is 0.5 in this case, which is pretty high. If you look at a good case here in the second example, you can see there isn't really any spikes in the uncertainty that we've been able to quantify from deep learning, but there's only a little bit around the edges, and that's because the edges are where you have the most decisions to make about what is clot and what is brain, right? Um, the other really interesting thing in terms of how the implications for the clinic was that the uncertainty metric was actually correlated with the dissimilarity coefficient. So basically, we can use this uncertainty metric to then flag cases where the segmentations weren't as good. And so that was very encouraging because now we have the ability to have a module where we kind of report this and flag certain cases in our hypothetical pipeline that could require intervention and manual correction. And so based on this, my postdoc and I have created a new technology. We basically created a prototype version of that software system that I was talking about several slides ago. And we're in the process of creating a startup company that we call CerebroSite. And basically, the prototype that we've been able to build, takes in CT images just like we were talking about, has image segmentation modules where we can segment and correct this. It will then read out the radiomics in this third step here and provide an image report for the clinician. And then based on all the data that it inputs or has been outputting and other data from the patient, it will then make predictions. Here's just 4 examples, but we have machine learning algorithms that are predicting the clot composition, stroke etiology, first pass effect propensity, final reperfusion, the patient's neurological status at 90 days, that kind of thing. And so with that, I would like to acknowledge my clinical partners who help collect these samples and collect these cases every day, my collaborators, my lab, particularly Dr. Tatsat Patel, who's the main driver of this AI work, and then others who are helping collect these cases. Thank you very much.
Interesting. So in terms of those experts not being able to locate, so is it that those visual indicators are confusing? Like, I'm not sure, is it the extent of the region of the clot, or is it even whether that's truly a clot or not?
Yeah, so I would say mostly the extent, based on the symptoms and the CT angiography, it is pretty clear kind of where the clot is and that the person has a stroke. I think when it comes to is if the clot isn't hyperdense, like if you can't see it on the non-contrast CT, the CT angiography only tells you where the clot starts, but not where it extends to, right? And so if it's not hyperdense, then that becomes very difficult to see. And, if it's close to a bone, then the bone will also interfere with all the signal that you're getting. And so I think it's mostly if it's in those regions near bones or when it's not hyperdense, there's a lot of disagreement on, where the clot is extending to.
So in terms of your end prediction of the prediction is the likelihood of a stroke?
Well, that's the thing is like, so once we're able to get this volume and calculate the radiomics features from that volume, we've been able to publish papers that show it can assist in predicting all sorts of endpoints. So there's prediction of procedural success or, the patient's neurologic improvement and, many different endpoints.
What extent is the extent of the clot significant indicator for your endpoints? Being able to predict that accurately.
Well, I think that's a good point. I think it's not only the extent of the clot, but it's also being highly accurate with that region because that is what dictates all of the radiomics parameters, right? if you have disagreement in the volume or you're accidentally including brain tissue in that volume, then it's going to throw off the calculation of those types of features. So that is the
I just wanted to make a comment as someone who's had a family member, close family member, have a stroke and have being able to maybe predict what treatments might be, what is going on with them is extremely invaluable. And I thank you very much, because even to this day, my family member is still struggling. So
I'm
this
sorry to
gives
hear
me-
that.
yeah, this gives me hope. So thank you.
Thank you.
Thank you for the presentation. I have a two-part question. First, when you were quantifying the uncertainty in segmentation of the plot, you were using U plot, which is a metric from like a paper published in Nikai 2022, I think so.
No, that's a metric that we came up with.
Okay, I just wanted to know that is the mod like the model was able to predict the uncertainty in the prediction, right?
Yeah.
So what will be the next course of action? Like how will the model tell the practicing user that it is uncertain, and to what degree it is uncertain in that?
Yeah, that's a great question. And that's like the rest of the paper that I didn't present. So basically, yeah, we then did a whole kind of AUC analysis to basically find what is that metric U clot, what is the threshold at which we should be then triaging those and sending them back to the clinician. So we did come up with like, oh, I think it was like 0.3. Right. So in the two cases that I showed, one was clearly over that and one was clearly under that. So that second one where it was pretty good, it's just like the border was a little fuzzy, that would have passed. Right. But then the other one would have been sent back and had some manual correction.
And the data set used for training were 250 patients, am I right?
Mm-hmm.
Patients, is it enough for the training of the model?
Yes and no. I think the biggest thing would be getting more for generalizability, right? When it comes to the medical imaging, we're chopping these down into very small boxes and doing training like locally there and then doing it across the whole thing too. So like, while we only have that many cases, in terms of like the ROIs that is training on is like thousands and thousands. Right. But I think in order for it to be generalizable, we need to start obviously getting much more samples from other centers, from other CT machines, that kind of
I
thing.
mean, yeah, I'm sure you have might of think out of it, because for the ischemic stroke data set and the riddling image segmentation, ample of images on physionet.org for that particular task. And thanks for the presentation. Thanks a lot. I was just thinking that can this particular network can be used for pulmonary embolism or DVT or something like that?
Yeah, we don't know yet, but yeah, we're hoping to expand. Yeah, thank you.
Thank you. So that's your small token for
Oh.
appreciation.
Thank you so much. Thank you.
There's no part. I'm sorry if I cut a lot. Speak. So you could turn it on. Still good. You get them now. Sure. You want to say something?
Hello, 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 three. Can you hear on the team?
So what was working?
1, 2, 3. 1, 2, 3.
Hello.
Hello.
It's only working through my laptop. So let's continue with my laptop.
Yeah, that's good.
Yeah.
Okay, presentation. So, right. You're sharing the slides and this. Wait. Hold on. So you can. Somewhere to your capacity.
Okay, thank you very much. So this is the presentation for the improving troubleshooting, problem-solving skill assisted with AI. Okay, so that's my name. So I work with I'm associate professor of teaching. I work with the Department of Engineering Education for the School of Engineering and Science. Okay, so this is the outline of my presentation. So the context, troubleshooting in AI, the result design, have some results, okay, key findings, and in the end, so discussion. Okay, what about troubleshooting? What is troubleshooting? So we can say that it's a cognitive process. Yeah, I just mentioned so this at the beginning. There's some kind of some misleading in the context of the science that troubleshooting is just following steps. And yes, that's okay, but it's following a step with some kind of background in terms of also the cognitive law that is related to this process. Identify faults, analyze symptoms, apply corrective actions. And so this troubleshooting gonna require the system knowledge and strategic thinking. Is related to the procedural and pretty much on metacognitive knowledge. So what about in terms of the AI associated with troubleshooting? Well, AI supports decision making, provides step-by-step guidance, and it's going to enable so the real times of feedback. So now in the industry, whatever is related to the power of the troubleshooting in the area of manufacturing, if we need to conduct some troubleshooting, so AI is going to support all these engineers and technicians so in solve these problems so in time. Okay, what about my research? So the project description. So this project introduced, I call this troubleshooting problem-solving instruction assisted with AI, TPAs plus AI, to improve so the troubleshooting skill and performance in the context of the course that I teach is electric circuits. Exploratory and control study will assess of the impact of this kind of instructions so versus the traditional instruction. Okay. So there are two phases. Okay, I display quite a lot of data, and so I'm trying just in the first part to conduct some kind of quantitative analysis and then some move to a qualitative. This phase number one is the quantitative approach. I have so this result question. Does this TPAs plus AI instruction improve student conceptual understanding and academic performance? How does this instructions going to improve so the student problem solving perception? And so then I was interested in see so the level of motivation so the students so when so this kind of instructions is going to be applied. Course description and participants. So I think so this course is electric circuits is for non-majors. So the amount of students that signed so the consent form so was 310. I think also in the phase number one, it's a control experimental groups. Okay, so when I had so the control experimental, they were assigned so randomly based on the section that they were enrolled. They didn't know at the beginning of the semester that's going to be so a specific instruction compared so with the traditional instruction. Okay, I apply so those instruments for the phase number one. The ECCE is the electric circuit concept evaluation. It's a concept inventory. Okay, to measure so the level of conceptual understanding of those students for AC and so these circuits. I have so this detect PSI, problem solving inventory, to measure so the problem solving perception. Pretty much of these instruments, so the idea is see about so the problem solving confidence, how the students so they feel confident in solving circuits problem, and also so the approach or the avoidance in terms of the it's more difficult or have to go for a different strategy, and so the personal control. Some kind of, so we can say, so the level of frustration. Problem is really complex. Now, when I stop here, I know that. So work with this problem. Okay, so the other is the IMMS. Okay, so the integrated materials, motivational survey. Two levels of the level of motivation based on these four criteria. confidence, attention, satisfaction, and so the relevance. How relevant is the content of this course for me or for my future as an engineer? So I have some of the exams and the chatbot transcripts. I asked students to provide for every problem that they were solving. So using chatbot, okay, so we use ChatGPT, provide so the copy of the transcript attached to the solution of the problem. So what about the intervention, the TPAs plus AI? Well, so we have some annual activities. In order to solve troubleshooting problems, first I needed for students to solve in some practical problems. In this course, there is no mandatory in the lab, but again, so I decided to include so the lab of these hands-on activities in the classroom. So we have so this kit, we provided some kind of AC and DC sources, tester, okay, oscilloscope, and so during the class time, so we conduct some kind of, okay, so activities, okay, so with, okay, so this student assisted with some TAs and student assistant, okay, during the process of the intervention. Okay, so what else? Okay, after so the hands-on activities, so we start working in this kind of problem. It's the typical circus problem. In some of the courses, and I can say statics, dynamics, second and third year in engineering. So what we do is, so this is the problem. Okay, those are so the parameters. Find the solution. Troubleshooting. This is the problem. This is the circuit. Something is wrong. You need to identify what is wrong in that circuit and provide so the solution. So as you pretty much, so you know, you are in the context of education. So we have so the structural problems and the ill structural problem. Okay, it could be 1 or 2 solutions or 3 solutions, depending on how the students can approach so this kind of problem. In terms of the prompting, solving so this problem, okay, I apply so this what we call so the Dexter method. The Dexter method is created so by okay, some scientists that they say, well, so we have so this kind of problem troubleshooting, so we have to follow so these steps to find the problem, consider so the causes, consider the solution, act and test and review. The challenging part is how when I translate all these steps into the prompt. But in the end, so we pretty much follow okay, so this method. And so that's one of the products that we apply so in this course. That's the data collection process. Was spring 2025, beginning of the semester, student science of the consent form 310. Okay, and so on the top you see the instruments how so they were applied. So the beginning, at the end of the semester, the pre and the post, and so the intervention. Okay, so we have hands-on activities in those specific weeks, and we apply so 3 different sets of troubleshooting okay, problems. Okay, so then I collected the data last year, I was here presenting so this methodology, but now I have some results. Demographics, okay, so the mean age is 21, 75% male, majority of the students are sophomore, multiple engineering majors, most of the students are from mechanical and aerospace, about pretty close to 70%. In the end, so the final sample after cleaning all the data, where the student that they complete so the pre, or the complete so the post, have another complete so the post, but no the pre's. The pre. Okay, final sample, so 187 for the control group. I have so 137 and 50 for the experimental group. Okay, so we decided to conduct the parametric analysis. Okay, so the data from continuous interval. Okay, we consider that this is appropriate, so we didn't run any Wilcon tests, but I think it's something that, yeah, so we can do just to double check that we're gonna follow so the parametric analysis. We use parity tests for the pre and post. For the groups, we run some independent tests. Moreover, we were that'd be interesting, apply some kind of ANOVA. So the alpha is 0.05. It's large samples, and we can say that it's kind of so robust study. For the electric circuit concept inventory, so we have some of the results. Okay. In terms of the control group pre and post, so no baselines of difference. You can see so the p-value is less than 0.001. For the experimental group, pre and post is also significant. Okay, and so we can say that it makes sense. The student at the beginning of the semester, we apply so the instrument, how so they manage so the concept of circuits, and it is supposed at the end of the semester, so there's gonna be some kind of difference. Okay, well, but then comparing so in the pre, so the control group and the experimental group, so we didn't find any difference, p-value 0.603, and so at the end of the semester, comparing so the pre and the post, we have so that is some kind of difference so between so the control and so the experimental group. Okay, seems like that's good in a certain way. So the key findings: no baseline difference, experimental group higher, post significant improvement in the experimental group. So then for the academic performance, I took all the information on the exams. There were 2 midterms, one final, and so I took also the information from the overall course grade. Okay, for the exam so number 1, so no difference between the pre and post. Okay, so the exam number 1 was applied on the week number 5. Okay, that's interesting. I think so it's gonna need to go over, see what happened in there. Okay, my favorite, I can say that was the first one. Okay, moreover, for the exam number 2, were some kind of differences between so the control and the experimental group, and the same for the final. The overall course grade is also significant. In the end, 70% of the grading was the same for the experimental group and the control group, but there was this 30% different. Okay, I'm gonna be some kind of cautious about so the overall course grade. But in the end, so the final exam is comprehensive. It's gonna cover content for all the semester. Key findings: no baseline difference, experimental group higher, post significant improvement. So the problem solving inventory, how they feel, the perception, how they feel confident in solving this kind of problem, how to manage so the level of frustrations. Okay, similar to the ECCE, okay, control group, pre and post. Okay, so we didn't find so well any significance of difference, but the experimental, yes. Comparing so the control group and the experimental for pre, no difference, but we found some differences in the post comparing so the control and so the experimental group. Key findings: no baseline difference, experimental group higher at post, and significant improvement. Okay, integrated materials motivational survey. We apply so this survey in the week number 5 and at the end of the semester. Okay, control group, pre and post, no difference. Experimental group, pre and post, we didn't find so any difference when we apply so the instrument. So when we compare, so in the pre, control, experimental, no difference, it makes sense. So at the beginning of the semester, seems like that there has to be same level of expectation. But in the post, okay, we don't find so any difference either. Okay, so that was the results. In terms of the IMMS, it's like we need to go more over. Okay, I just started running some kind of alpha Chromebook and then some factor analysis to see also the instrument that I was applying. This is not a common instrument to measure some motivation in the context of education. We pretty much apply some kind of the standard instruments of the motivated EMSLQ, that is most common, and I decided to go for these things. But it seems like in phase number 2, I'm gonna go into more deep. So what happens with instruments, also the instruments was with the process of internal reliability, because I went through the process of the content validity. Key findings: no significant differences, motivation unchanged. Okay, pre and post in comparing so experimental and control group. In the end, so those are also the results, key findings. No difference at the baseline, so the ECC increases the conceptual learning. Okay, in terms of the perception, the problem solving increases in the experimental group, performance, and so there were no changes in terms of motivation. And then so we have so the phase 2. Phase 2 is ongoing process. Okay, I just wanna share with you. So what I have for this phase, I'm planning to start so during the summer of this year. Okay, so I have so all this quantitative data. So we went to the runnings of the statistical analysis. Okay, what I'm interested in now is see what about inside of this chat conversation. Okay, analyze the chatbot interactions, identify effective prompting, inform instruction, and so the part of the assessment. Okay, I'm planning to conduct develop some kind of framework for the part of the coding. I'm interested to see about so the quality of the prompting. They are really challenging. So this chatbot, okay, how in terms of the surface, so they are so doing so this prompting. Okay, so the strategy for when they using so AI. So what about the cognitive engagement in this prompting? So they are just discussing about factual knowledge, but they are going into more deep with the part of the conceptual, okay, or moreover going into the metacognitive. So reflecting so with this chatbot in so the interactions and see how I can link all this coding from this chatbot conversation into, okay, so the quantitative analysis. Okay. I just want to say thank you to the Office of Curriculum Assessment and Teaching Transformation because they provide me so the support to collect so the data. So last year, as you see, so there were around 300 students. So we applied those instruments in class time. And so I needed a lot of resources, okay, TAs and student assistants to collect so this data. Thank you.
Questions?
I guess I had a technical question. So when the next stage you said was, to look at their prompting, right?
Yes.
Do you have a way that you're gonna do that to look at their chatbot interactions?
Okay, well, I'm still, okay, working with some collaborators trying to decide what is gonna be so the framework for the coding.
So will you be creating your own chatbot for this then, like with the?
Oh, well, no, actually, so we have some chatbot conversation. So when the students, so they work solving a problem, so using AI, okay, so they were solving the computer with chatbot conversation, use ChatGPT. Okay, so we ask the students to download the conversation, put it as a part of the assignment. Okay,
Just so you know, there are a couple tools out there that allow you to have students working in groups and have group chat folders, and you can monitor what they're doing. You can create an assignment. I'm using that right now in my course, and it links up to like the 5 main chatbots out there, like Claude, GPT, Gemini, Perplexity, and I always forget the last one. That's why I was just curious. And in terms of identifying affecting prompts, so do they get any training on like the different types of prompts, like a 0-shot prompt or a chain of thought prompt? Do they get any?
well, so what I can say, so in terms of the prompting, I saw one problem in class, and I tell students they have to follow so the way that I solve the problem. Okay, I have my own screen and say, well, so the first point is, for example, so I have some specific rules. So you don't have to, okay, take a picture of the problem and so no, you have to describe. And I give the instruction on how to describe, for example, the electric circuits. Okay, those are the instructions. Step number 1 is to listen to this, step number 2 is step number 3. Okay, and so then I start solving by myself. Okay, so using so the chat, and those are the instructions for them, their own problems, and also in the homework assignments. So with the prompting, okay, so I use this method, the DEXA, following so these steps. Okay, last semester I made some kind of improvements, and this semester I'm doing so another improvements. I think so one of the challenging parts in this development of this AI as part of the curriculum, as part of the instruction, is how the quality of how so they is going to be typing, so this, okay, the prompting, okay? And so I would like for this study to use some kind of generic AI or ChatGPT, I'm using so ChatGPT, Gemini, any other, because in certain way, I'm interested, so this instruction to be translated to any other course without any issue in terms of the resources.
The tool that I'm using is called Boodlebox, and one of the things it has is that it has customized chatbots by different disciplines. And I'm wondering, because I have a trial subscription to them right now, if they have anything that would be relevant, but you can make your own chatbot.
Yeah.
You have students interact with that as well, especially if you have like a step process that you want them to follow.
Yes, actually, that would be so the one of the following steps, because one of the challenging things, so specific in my course, is a for example, for define so the voltage in some textbook they use a V, in another textbook they use E. In order to avoid that kind of cognitive overload, that when we are describing so the power factor in some textbook they use FP, in another textbook they use F uppercase, P uppercase, completely different. So that's gonna be so one of the following steps, just to that this chatbot personalized, that's gonna go for a specific textbook with that specific way to describe. So those, because in the end, so that's gonna create students of this kind of cognitive overload. So, yeah, thank you.
Thank you, and let's thank Presentación again. It's also his birthday today, so happy birthday. So we have some, so we look at state from the what kind of the building this so these are very students. So for first off, there is a sort of connection. So we're using working data and also we have this presented conference interaction. Last no. Okay.
Yeah,
We have
So I'll just.
not only does this give you the predicting, but also gives you some.
Yeah, so
you have that like a single.